Hello, my name is Michael Lynch. I'm a PhD student at the University of Limerick in Ireland, and today I'll be showing you some of the work we've done on supervised demultiplexing of single-cell RNA sequencing using cell hashing and SNPs. So I'm going to give a quick introduction to demultiplexing. What is it and why is it important? How does demultiplexing work? What are some current limitations? And what we've done to improve on these. So first off, what is it? Why is it important? So let's take a step back for a moment and look at non-multiplexed or single sample, single cell RNA sequencing. So this was limited to sequencing fewer cells with an increased cost per cell. So in the droplet encapsulation step, the aim is to encapsulate one cell and one barcode in each droplet, and this is called a singlet. However, it's a stochastic process, so we don't always get a ratio of 1:1:1. We can also get an empty droplet, whereby no cell has been captured, or a doublet or multiplet, where two or more cells have been captured in the same droplet. As we load more cells onto the lane, the rate of doublets increases, and this is problematic because these can confound downstream analysis. If multiple biological samples need to be sequenced, they have to be sequenced on the separate lane. Multiplexing then, it allows sequencing of more cells at reduced cost per cell. So why is this? When we multiplex, we pool cells from multiple different biological samples together prior to sequencing, and they all get sequenced on the same lane. So as before, we have singlets and empty droplets. However, we now can have two types of doublets. So we can have a single sample doublet, where both cells are from the same initial biological sample, or a multi-sample doublet, where the two cells are from different biological samples. So as we increase the number of groups being pooled, the ratio of multi-sample doublets to single sample doublets increases also. And this is important when we come to demultiplexing. So the demultiplexing then is the assignment of cells back to the original sample and the identifying of doublets, specifically these multi-sample doublets. If we demultiplex correctly, we get a reduced cost per cell, or alternatively we can sequence more cells for the same budget. We'll also get some reduction in batch effects. However, if we don't demultiplex correctly, we'll get a reduced statistical power, wasted data, a reduction in log fold change when looking at differential expression, and in the worst case scenario, misleading biological interpretation. So our aim is to maximize the number of cells we can assign, specifically to the correct group. So how does demultiplexing work, and what are some of the current limitations? The main method used for demultiplexing is cell tagging or hashing, which is a combined experimental and computational approach. Here, cells are tagged with a sequenceable barcode using either HTO, CMO, or LMOs prior to pooling. When the samples are pooled, however, we get non-specific binding. So some of these tags detach from the original cell and reattach to a different cell. And this gives us this bimodal distribution that we see here at the bottom left, where we have an upper signal distribution and a lower background distribution. Looking at the expression of the different hashtags on the different cell types, for a singlet, we'd expect to see strong expression in one of the groups, for example, the hashtag 2 group here at the top. For a doublet, we'd expect to see strong expression in 2 or more groups, as we see here for the hashtag 1 and 2. However, we can also get a hashing negative. In this case, a cell has indeed been encapsulated in a droplet. However, for experimental or computational reasons, we're unable to assign it to a group due to its hashing quality. So we expect the performance of hashing demultiplexing algorithms to be dependent on the hashing quality. So here we've simulated some hashing data for 4 groups, hashtag 1, 2, 3, 4, as well as some doublets and negatives. We can tell it's quite good quality data because we see very good separation between the signal and background distribution. We applied HTO demux, a popular demultiplexing algorithm here, and we see really good precision recall across groups 1 to 4 in the doublets, but to a lesser extent the negatives. But overall, very good performance. We then simulated some lower quality data. So here we made some changes. Overall, we've reduced the signal to noise ratio, as well as varying the group sizes. So we can see hashtag 3 is a larger group, hashtag 4 is a smaller group. Again, we applied HTO demux, and we see significantly poor performance here. We see poor recall in #3, poor precision in #4, and very poor precision in the negative group. This result for the negative group here indicates that there are lots of cells that we haven't been able to assign because of issues with their hashing quality. And we want to avoid this if at all possible. We saw some of these issues with hashing quality on a recent data set where we had 10x single cell data from CD4 positive immune cells. Here we had 6 donors and we were comparing between primary metastatic tumors. So it was very important that we knew which cells came from which person. If you examine our hashing distributions, we see in #2 we have very low signal. In #3 and 5 especially, we see a very low signal to noise ratio. And in #5 we see that the signal has been shifted down a little bit. So when we ran different demultiplexing, hashing demultiplexing algorithms, we saw very different results depending on the algorithm. So that brings us to our next method for demultiplex. Natural genetic variation can also be used to demultiplex between genetically distinct samples. So here the aligned reads are compared to a reference to determine whether a SNP or a single nucleotide polymorphism is present at a particular location. However, these methods can be compounded by sequencing depth, ambient RNA, having short read length, cell type heterogeneity, or the presence of mutations. So visualizing this data can be quite tricky. So to explain to you what we have at the bottom, our rows are SNPs and columns are different cells. So the blue here represents 0,0, which is no reads support the SNP. 0,1, there's reads to support the SNP on the alternative. 1,0, there's reads to support the SNP on the reference. And 1,1, there's reads to support the SNP on both. So as you can see, it's very noisy data. Doublets here can be identified if a cell or a doublet cell contains SNPs from 2 separate groups. So we applied supercell, a popular SNPs demultiplexing algorithm, to our 6 donor data set with poor hashing quality to see how it compares to the hashing algorithms. Let's look at the cells highlighted in green first. So in #13456, and for some of the doublets we see pretty good agreement. However, if you look at the agreement between the supercell #2 and the HTO demux #2 groups, we see only one cell has been commonly called between them. Although if we look at the column for supercell #2, we see almost 1,000 cells have been called in this group. So what's going on here? If we further interrogate the supercell hashtag group, we see that there's HTO expression being picked up in multiple different groups. For example, we see it in #3, 4, 5, 6. So this indicates that this isn't in fact an actual #2 group, and it is more than likely the doublets that HTO demux picked up. So why does this happen? Well, it's been reported before that these genotype-free SNP methods can sometimes miss smaller groups. So what can we do to improve on this? We like to visualize things, right? So we took the 1,000 genomes common variance list and subset. We subsetted to SNPs seen in genes commonly expressed across most of the cells in our dataset. We then called SNPs in each cell and further filtered that down to SNPs actually seen in these donors. And this is what we got. Here we have 3 possibilities: if the SNP is absent in our cells, if we've no reads to determine, or if the SNP is present. So we can then split this data out by the different hashing algorithms results to QC. So first, let's have a look at this HTO demux #2 group. It's a pretty small group. However, if we look closely, we see something interesting. On the left, we have a main SNP profile. However, on the right, we can see a secondary, potentially tertiary SNP profile. So this shouldn't be there, and it's potentially indicative that our cells here from one of the other groups. Now let's have a look at this negative group. So there's over 2,000 cells in this group, which is over 10% of our data set. So these are cells that weren't able to be assigned because of issues with their hashing quality, not their RNA quality. I would ideally like to be able to assign these back to a group. So if you look closely, you can actually see a lot of these negative cells. They look pretty similar to that hashtag 5 group, don't they? So maybe there's a way that we can systematically reassign these. So far, we've filtered down the SNPs, and we've called the SNPs in each cell. And that gave us the heat maps that we visualized in the last couple of slides. So if we want to move on to reclassification, we know we can call some cells, just not all cells, using the hashing methods. So we take the cells that we can confidently call and use their labels as training data using their corresponding SNPs as features. We can also simulate doublets by combining singlet SNP profiles from these high confidence cells. We can then predict on the remaining cells using the SNPs as the features. And these are the cells that we couldn't confidently call using hashing methods. So we've implemented this workflow in an OR package that we're calling demuxSNP. On the right, we visualize PCA of some simulated SNP data. On the top, we have the training labels, and the bottom, the predicted labels. As you can see, the predicted labels fall in a similar area in SNP space as the training labels. So when we apply our algorithm demuxSNP to the data we saw earlier, what do we get? So let's first have a look at this HGO demux hashtag 2 group on the left. So on the top, we've colored by the HGO demux assignment, which was hashtag 2, and we split it by our demuxSNP assignment. So as we suspected, about 2/3 look like they actually were hashtag 2, and this is confirmed with the demuxSNP assignment. And we can see here on the right that about 1/3 have indeed actually been reassigned, mostly to hashtag 5, but also a couple of different groups. And we can see a distinct SNP profile in each group. Now let's look at the negatives. So to remind you, these were cells that the HGO demux algorithm couldn't assign due to issues with the hashing quality. So all these cells on the right, HGO demux has called it as a negative. And below this, we can see that now they've been assigned to various different groups by demuxSNP. The majority of which going to hashtag 5, which is consistent with what we expected earlier. So now let's look at some benchmarking. First, we want to assess demuxSNP's ability to assign singlets in a heterogeneous tissue. So first, we isolated high confidence singlets from our dataset to use as ground truth. So this gave us about just over 8,000 cells. Looking at the bar plot at the bottom, we see this very varying group sizes. We see the hashtag 2 is a very small group, hashtag 5 and 6 particularly large groups. So we then performed a 10-fold cross-validation, and we varied the proportions of training to test data. So over on the right here, we can see consistently high precision recall in each group, with only minorly poor performance in the hashtag 2 group, which is particularly small. Additionally, we don't see a massive change in performance when we change the proportion of trained to test data. So in practice, we'll usually have doublets in the data set. So we wanted to assess the ability to assign these multi-sample multiplets in the same data set. So taking these high confidence singlets, we swap barcodes to simulate 20% doublets. Again, we performed a 10-fold cross-validation, trained on 20% singlets, and simulated 1,500 doublets. Again, we can see a slight reduction in performance. However, we're still getting consistently high performance across each of the groups. There is a slightly lower recall in the double assignment. However, we expect we should be able to assign the remainder of these doublets using downstream doublet detection tools. Now to look at some limitations. So we expect to see poor performance when we have a lower sequencing depth or fewer expressed genes. So why is this? When we filter our SNPs, we're looking for SNPs in genes commonly expressed across their data. However, if the sequencing depth is low or we have fewer genes being expressed, we see fewer genes which are expressed in almost all cells. And this is plotted here on the bottom right. So we can see it drops off pretty quickly. So this gives a trade-off between including more SNPs in our data with partial information or including fewer SNPs which have better coverage. And this is reflected in the performance. So we see poor performance in both singlet and double assignment here. However, we do expect there to be some usefulness of this algorithm on this type of data, albeit with added caution and inspection of results. So to conclude, with cell hashing, we can confidently call some but not all cells. With SNP methods, there can be some limitations, for example, with small group size. So with demuxSNP, our algorithm, we can identify cells miscalled by other algorithms, and we can assign miscalled and uncalled cells using a supervised classifier. So the code for this is on the bioconductor development branch, and that can be accessed through this QR code here. Thank you for your attention.